We are preparing for the SeekOne DD single-cell multiplex ligation-based probe sequencing experiment. After completing MLT hybridization and washing, we now proceed to droplet generation for single nucleus barcoding. Equilibrate the barcoded beads to room temperature for at least 30 minutes. Before use, equilibrate the 10 times probe ligation buffer to room temperature until fully dissolved. Make sure no visible precipitates or crystals remain. Thaw reducing buffer and primer PR on ice. Vortex the prepared reagents thoroughly before use. Add the probe ligase mix. Briefly centrifuge all reagents together and keep them on ice until use. Use the probe ligase mix promptly after preparation. Place the instrument on a stable bench, power it on, and run the self-check. Now let's calculate the nuclei input for droplet generation. The maximum loading amount is 24,000 nuclei. Because the capture efficiency is roughly around a half, your target capture number should be adjusted accordingly. If your goal is to recover about 10,000 nuclei, you'll need to load roughly 2 times that amount. The total volume is 80 microliters. 22.3 microliters are occupied by the master mix, so the remaining 57.7 microliters must come from nuclei suspension and PBS-RI combined. First, calculate how much nuclei suspension you need. For example, if your concentration is 1,000 nuclei per microliter and your effective loading target is 20,000 nuclei, you would add 20 microliters of nuclei suspension, then subtract that from 57.7 microliters to determine the PBS-RI volume. In this case, you would add 37.7 microliters of PBS-RI. Now prepare the reaction mix on ice. Add the reagents in the order: probe ligation buffer, probe ligase mix, reducing buffer, primer PR, and finally PBS. Pipette 15 times to mix. Spin briefly and keep the mix on ice.